Giacomo Ramos asked me to provide a brief review of new breakthroughs in imaging technologies. And well, in the past, I would say decade and a half, there have been so many revolutions in these technologies that necessarily this review will be incomplete, but hopefully will provide plenty of discussion during the next two days about what will be the important technologies that we'll need to use in the near term because they're ripe for lifetime, and which are the technologies that is worth investing for the far future. And so one main objective of lifetime is to be able to retrieve the transcriptome and the epigenome of each single cell in a tissue and during the progression of diseases. These kind of measurements were traditionally performed using ensemble sequencing technologies. And these technologies have a main problem, and that is that they usually require the mash together of dozens or millions of cells to have a single output. And so that means that they're unable to dissect heterogeneity. So to realize this objective, we need to rely on single cell detection. And we need these technologies to provide us with certain features. So we need specificity in order to know what we're looking at, what species we're looking at. We need to be able to detect many species at the same time. We need, of course, to retrieve or conserve the spatial information. This was stressed already before. And we need to retrieve the dynamics of the system. So the first two features are typically associated with sequencing-based technologies, whereas the last two are most of the time the realm of imaging technologies. But as you will see in my talk, and I think in all the talks of this session, these boundaries are very quickly starting to blur. So possibly the most important feature of an imaging technology is that it allows us to see. It allows us to see how cells are organized in tissues, and it allows us to look at internal organization of cells. However, this ability in the past has been impeded by many limitations. So one of them is a lack or insufficient spatial resolution. The second one is the existence of the optical aberrations. And the final is a limited number of colors that we can visualize with a conventional microscope. And I'm going to show you today that there have been many revolutions in the last decade or so that have uplifted many of these limitations and now can allow us to actually address all these features. And so the main limitation is the lack of resolution. And in practical terms, what this means is that we cannot really look at the structures of complexes that are smaller than the diffraction limit of light, that is about 250 nanometers. This problem has been solved in the last decade and a half by a number of technologies that are called super-resolution microscopies, and that now allow us to reach the tens of nanometer scales. And these technologies have already been used very often to solve very important biological problems. An example is the realization that nucleosomes are organized into nucleosome clutches. Another kind of study recently showed that topologically associated domains exist at the single cell level. And further advances in these techniques are now allowing us to even reach the nanometer scale to resolution. So in parallel to these developments, in optical microscopies, there have been also revolutions in electron microscopies. So now with electron tomography, for example, we can look deep inside cells, have amazing nanometer scale resolutions with lower specificities than with optical microscopies, but these allow us to directly segment the different compartments and different structures within cells. And this has been recently used, for example, to directly visualize DNA and nucleosomes, and then look at the different structures that the chromatin fiber takes in a cell. And also very important developments have been made in the field of cryo-electron microscopy. And so now we're able to solve the structures of very large molecular complexes at sub-nanometer resolution, so Armstrong scale resolutions. And you may be asking yourselves, why do we need such big resolution? So imagine in the future if we're able to solve these kind of structures with this kind of resolution, but in the context of a cell within a tissue, then we should be able to retrieve stoichiometries of complexes and architectures of complexes in normal tissues and see how this change during disease progression, and then possibly even design drugs that will block the formation of disease-associated complexes. So the second limitation is the existence of optical aberrations in the system, and this has many consequences. One of them is that it prevents us from being able to see very well in depth in tissues, and also from monitoring the dynamics without a toxicity in tissues. So these both combined mean that we have a limited ability to monitor the real-time dynamics deep in tissues and organisms. And so these kind of problems have been tackled, and this was previously mentioned in the previous talk, by two technologies, light sheet microscopies and confocal-based technologies. These both techniques now provide with a very high optical sectioning capability and are able to image specimens at much faster temporal resolutions. And so I'm just going to show an examples of this. The lattice light sheet microscopy developed in the Betzek lab that now has subcellular optical sectioning capabilities so that we can now follow the dynamics of intracellular structures with very low phototoxicity and very high time resolutions. We can also use it to look at complex tissues, such as in this case, the zebrafish eye. We can segment each cell in the tissue, blow it up computationally, and look at the morphologies of each of the cells in the tissue. And then we can do this game over time. So that means that we can now characterize, you'll see a bit later, the cell dynamics within tissues, how cells are moving within tissues, and we can even look at the stages of the cell cycle to which each of these cells is. A second family of technologies, now based on confocal, has been also recently used to monitor the real-time dynamics of processes in living organisms. And a good example of this is transcription in living Drosophila embryos. And so in this case, we're looking at nuclei that are very quickly, you'll see, undergoing mitosis synchronously. They go back into T1, and hopefully you will see all these green bright spots. Each of these spots represent a nascent transcript being produced by the cell. So if we monitor now the signal of these spots, we can now have a trace of how transcription evolves over time, and so we can understand the mechanism of transcription of this gene. By looking at what cells are transcribing and which are not, we can directly measure the heterogeneity in the sample. And by looking at the timing of transcription, we can understand the synchronization mechanisms. This has been recently pushed one step further by the detection not only of the nascent transcript, but also of the regulatory elements that are important for transcription. So in this case, for example, the group of Thomas Gregor has been able to detect the nascent transcript and the position of the promoter and enhancer, so that they have shown that you need to have contact between the enhancer and the promoter to start transcription, but you don't require them for elongation. And so the final limitation is our ability to see in multiple colors. And this arises in part due to the lack of enough or good enough labeling strategies. Also comes from the fact that conventional microscopes allow us only to see in a very limited number of colors. And so in practical terms, what this means is that very often we can see things with very high degree of detail, but we don't really know what we're looking at. So this problem has been tackled in the last, I would say, 4 or 5 years by a number of technologies that try to increase the specificity of labeling and the multiplexing abilities of microscopes. And so one such example is MERFISH. MERFISH relies on the combinatorial detection of multiple RNAs in the single cell level. And because this is an imaging technology, we can look at the single cell in the context of the full tissue, and so we can preserve the spatial information. This technique allows for detection of hundreds to thousands of RNA species in each single cell with very high efficiency and with very large dynamic range. So we can detect from 1 to about 1,000 single molecules per cell. And so with this, we can build single-cell tissue atlases with spatial information conserved. We have recently developed a variation of this technology that does not rely on combinatorial labeling, but rather on sequential labeling that we call Higham. And these allow us to retrieve the 3D organization of chromosomes in intact entire organisms. And so for this, what we did was to immobilize a number of Drosophila embryos on a microfluidics device. We coupled this to a microscope so we can visualize each cell in the embryo and in each embryo. And now we introduce probes through the microfluidics device so that we can target a specific genomic locus. We image the 3D position of this locus in each cell, in each embryo, and then we photobleach it and move on to a next probe that will now label a second locus. We photobleach this and we move on to the next probe. We do the sequential labeling and imaging n times, and so at the end, we get the 3D positions of n different genomic loci in each cell and in each embryo. If we couple it to the detection of transcription, of specific genes in each cell, now we can correlate directly the transcriptional status with the conformation of DNA. And so similar variations of this technology have in parallel been developed by the Bettinger lab, by Chao Wei's lab in Harvard. And so how can we benchmark this kind of technology? So the way we did it is to compare the metrics, the contact map metrics that we obtained with Hi-C with that obtained by Higham. And you can see already here there is a very good resemblance between the two matrices. But Higham does not only provide us with contact maps, it also provides us with distances. So we can build directly pairwise distance maps. And with these, for example, we can directly see the appearance of topological domains. But these still are ensemble measurements. I told you we can get access to single cells. These are the pairwise distance maps for all of the cells in a specific embryo. And we can blow up any section here, and we can directly dissect the heterogeneity in chromosome organization. And you may be asking yourselves, what are the sources of this heterogeneity? So an obvious candidate is obviously transcription, right? We know at this stage embryos, there are several presumptive tissues that are being formed, each of which will be expressed in different set of genes. And so presumably, if we group together the cells that are in the ectoderm, the endoderm, and compare to the chromosome organization of the cells in the mesoderm, they should be different, right? And so to do so, we added the labeling of a transcript of a gene that is called SNAIL that is expressed specifically at nuclear cycle 14 embryos in the mesoderm. So now by looking at the cells that are expressed in SNAIL, we can segment them, and we can group them together and get chromosome structure for the cells in the mesoderm, and we can do likewise for the cells in the ectoderm and in the endoderm. And you see straight away that they have very different chromosome conformations. So from this, we can conclude that chromosome organization changes during transcription activation, and you can imagine similar techniques being used in tissues, where we can now correlate the organization of chromosomes and the network of interactions between promoters and enhancers and the output, which is transcription. And so you might be asking yourselves, is this enough? Do we have the tool set that we need to tackle these issues? Are there future challenges? And I think there are important challenges. I told you that we can now retrieve a chromosome organization in 3D. If we do this over time, we can get a fourth dimension. We can couple it to transcription, so we now have a fifth dimension. But this was mentioned before. We are not talking about the methods that detect proteins. There have been many important developments in single-cell proteomics, in mass spec imaging, in sequential images of proteins, as was shown in the previous slide. So if we can bring these detection methods on board, then we have a sixth dimension. And of course, there's many other species that will need to be imaged to have the full picture. We have all the different kind of metabolites, lipids, and glycomes in the cell. So this will bring us to an n-dimensional imaging, where we will be able to actually have the full picture of the ID card of a cell. So the second challenge is multiscale imaging. I told you that we have unprecedented details of structures of proteins at nanometer, sub-nanometer scales, but most of the times, currently, these are obtained in vitro, or if we're lucky, in single cells. But imagine what we could get if we were able to realize these measurements in the context of a tissue, to look at the immediate neighborhood of cells, or even at the whole tissue. This will allow us then to, for example, see how specific changes in a cell are propagated to the rest of the tissue, or how tissue homeostasis drives the fate of single cells. So we need to be able to retrieve multiple scale spatial information from nanometer to centimeters. And I'm just going to show you a teaser of a similar experiment that we have recently done in a completely different system. Here we have setting up a fight between Myxococcus colony, here round shaped here on the left, and an E. coli colony on the right. They're going to fight. One of them is going to win. And so I'm not going to keep the suspense very long. The winner is going to be Myxococcus. Myxococcus is equipped with specific devices that allow it to move very quickly on surfaces collectively and attack E. coli cells, and also has devices for killing E. coli that essentially rely on the penetration of the membrane, depolarization, and then absorption of the nutrients that are produced by E. coli. So to study this mechanism, we need at the same time to be able to look at the full picture, both different colonies, but also to have molecular detail. And so if we do this multiscale imaging, now we can zoom in, as in Google Earth. We have the full picture, and we can look at the, for example, formation, of complexes that are driving the killing of each of these bacterium. And so we can, of course, then come back and look at what is the role of each cell in the context of the whole predation process. So with this, I want to finish here. I told you about a number of disrupting imaging technologies. As I told you at the beginning, this is not a comprehensive review, and there's probably many techniques that we also need to take into account. It is very important that these are combined efficiently, complementarily, and synergistically with single-cell sequencing technologies, and that both are adapted so that we can essentially look at relevant model systems, such as organoids or tissues from patients, and that we do a lot of work in order to build optimal data storage services, AI-enabled integration and modeling so that we can understand the output of these measurements. And with this, I want to thank the people that have done the work that I've shown you today. I want to also thank financial sources for their support, and I want to thank you for your time.
Thank you very much, Marcelo. There are questions. Don't be shy.
Yes.
There's one over there.
Thanks, Geneviève. Very, very exciting things. I'm wondering how you would tackle the integration of, let's say, RNA-seq and the imaging beyond just getting better labels for clusters. So do you envision some sort of more deep way of potentially also finding something like networks, and if yes, how to?
I have no idea. No. I think this is one of the objectives of this meeting, starting to set up working groups so that imaging people come together with the single-cell sequencing people. They each understand the formats, the kind of measurements that are being made, and how can we, at the same time, integrate the data, make sense of data, store it. I don't think we have enough
So it's really
information.
decoupled still a bit, right?
Yes, I think it's very early.
Hi, this is more a comment. So we can view images at any resolution. We can see a lot of things, but at the end of the day, we don't know to tackle morphology, especially in the context of a complex disease like cancer, because we only have in our hands the concept of pathology, basically. So you go to the pathologist to describe the shapes in a very qualitative way. And I think one of the things that we have to do is to develop some quantitative concept to tackle those pathology if we want to go beyond the clusters of cells and things like that.
Definitely, that's a very good comment, yeah.
Well, this is, of course, raises the issue of the various scales that we need to be able to integrate. So, for sure, important.
Regarding your C. elegans embryo imaging, beautiful imaging data, when you cluster these regarding to the really individual images, I don't know if you have done this, but how many different states do you see?
We don't know yet. This is something we're doing currently. I mean, obviously the holy grail here would be to try to see if we can get predictive transcription information from only the genome organization, right? So can we take directly different cells and cluster them? And it's not trivial at the moment. So there is a lot of heterogeneity even within presumptive tissues. So if we look at all the cells within the presumptive tissue, we see a very large heterogeneity. And the heterogeneity, even though it's smaller than what it could be if we take the whole embryo, is still important, right? And if you think about it, it's maybe not surprising, right? Because, I mean, when you produce specific transcript that we're detecting here in the cytoplasm, this mature transcript, doesn't mean that the process of transcription is going on at that exact time, right? It could have passed minutes ago, and so that means that the chromosome conformation that you're looking at in this snapshot, right, is a few minutes after the transcript got made. So it is not surprising that there is heterogeneity and that you'll be partially predictive, but probably not fully predictive from just genome information, genome organization information.
Well, the beautiful picture you were showing about this multiscale resolution and the analogy with, for example, cosmological structures possibly allows the modeling of this information with physical approaches and physical laws regarding reaction, diffusion. Are you already working on this? Because this would be very fascinating for our physicists.
So, no, at the moment we are working on getting good data. We haven't, the avenues for modeling are huge because you can think about modeling inside cells and looking at diffusion, etc. But these cells are having directed motions. The motions are directed essentially by nutrients availability in the neighborhood. And so you need to essentially use cell models, cell automata models to try to understand these and build in behaviors of the cells. We think that each of these cells has a different transcriptome, so there is cell types within this colony. And so obviously that's the other avenue that we're looking into. Yeah. Thank you.
Yeah, actually, if I can add a little comment about it, I think this brings the attention to the importance of having some very well-defined systems to study that are sufficiently low, even if it's already a huge complexity, but sufficiently low complexity. So I think that's going to be very good training systems to tackle much bigger problems. Because of course, here we're talking about human health, but we have 40,000 billion cells at the end of the day, and I think it's important, like studying organoids in a very well-defined conditions, but even studying like model systems, like simple animal development, that can be very important. Because even if you have multiplexing technology, you can study hundreds of points of components, it's still, there's still a huge source of variation. So that's going to be one critical part of the project.